Alrighty. Hello, everybody. What's up, Ethan? What up, Not Oster? Darpan, why you've been waiting for 2 hours? I don't know why you'd be waiting for that long. What's up, Alicia? We're just starting it up here. Things are just getting rolling. Things are just starting. What's up, Ishan? Aristotle gang. Aristotle is that your real name? Aristotle? Aristotle, that's a cool name. Alicia, will the bio teachers be there for tutorial? Good question. I don't think they all will be. I'm gonna try and sneak out of our meeting to help some people with during tutorials, see if we can take a break at that point in the day. But I can't say that's true for all teachers. Cannot make that promise for all teachers. I don't know. Not sure. What's up, Prabhav? How you doing, man? Darpan, I can't answer any of the A-level questions because remember those A-level questions are a list of questions, one of which will be selected for the exam. So I can't answer those questions. Just giving you a heads up on like what kind of topics to prepare for and kind of get you ready, but I can't answer those specifically because that would defeat the purpose of releasing them. I would be giving you the answer. Inia, can I explain ionic and covalent bonds? I can. You don't really need to know too much for this test, to be honest. The three types of bonds that you should know between those three. So ionic is a bond that forms between two atoms. It's usually a pretty strong bond that connects two atoms. It's where they do not share electrons, and it's between something like a sodium and chloride, like a chlorine atom when you have salt. That would be an example of an ionic bond. Most bonds that we're talking about when we talk about biomolecules are going to be covalent bonds. All the bonds between the carbons in things like sugars and fats and proteins, those are all covalent bonds. And they share electrons with each other. And then you have hydrogen bonds, which are the bonds specifically between hydrogen atoms that are very, very weak. And we typically talk about those when we think about water, because that's how water molecules attract to each other with hydrogen bonds. What's up? What's up, Ananya? Aristotle, that is your full name. Cool. That's awesome. I like it. Aristotle. So with the whole hypo versus hippo, or sorry, hypo versus hypertonic, that thing, let's see, what are some people saying as a way to remember it? What's up, Akash? Darpan, you're saying hype equals hippo. Since hippos are fat, fat equals getting larger, so hypo equals getting larger. See, the problem with that is it doesn't mean it always. It's a comparative term, so it would depend on which solution. So, the way you can think about it for me, the way I remember it is hyper, right? Like if someone's hyper, they've had a lot of sugar. Like right now, probably a lot of kids out there that have trick or treated their hearts to their heart's content, and they filled up their pillowcase full of just processed sugar. I'm talking just foot deep of straight up candy. They're going to get a sugar rush, right? They're going to get hyper. So they have a lot of sugar. So they have a lot of, so when I say hyper, I think of like hypertonic solutions have a lot of solutes like sugar or salt or other things that are dissolved in the water. So a hypertonic solution to another solution, if you have two solutions, the one that's hypertonic to the other one is the one that has more stuff in it. It's hyper, it's got more sugar, more salt, more anything in it. The one that has less solutes is hypotonic. That's the way I remember it. And then remember, if you have a hypertonic and a hypotonic, water will always move from the place that's hypotonic in terms of the solutes to the the side that is hypertonic with the solutes. Maybe I'll show you a diagram just to pop this one up. See if we can get a decent image here. So when you think about cells in particular, yeah, so this one, they took away my button that I used to like. So hypertonic, there's more stuff in the solution than in the cell, so the water moves out and it shrinks. Hypotonic, there's more stuff in the cell than outside, so the water moves in and it swells. So I guess you could think of a hippo in that sense if you're talking about a cell in comparison to the solution. Not a bad one. Not a bad one. Hope that clears up. What's up, Mr. Chang? Nice to see you. Akash, nice to see you. Renee, hey. Anna, nice to see you. What's the difference between the cell membrane and the cell wall? The cell membrane is all cells have a cell membrane. All cells have a cell membrane, right? Whereas only plants. It took away my view image button. There used to be a button here that I could click where it just said view image, and now it's gone. So now every time I click on an image, I can't, it just takes me to like a web page. So all cells have a cell membrane. This is what's made of lipid. It's the lipid bilayer, the phospholipid bilayer. It's where you have protein channels and protein transporters, and you also have things like endocytosis and exocytosis happening. Cell transport is happening across this phospholipid bilayer, which we call the cell membrane. Sometimes we call it the plasma membrane. But both plant and animal cells have this. The cell wall is this yellow ring around the outside of the plant cell. And it's made not of phospholipids. It's not a lipid bilayer. It's actually made of cellulose, which is a type of carbohydrate, actually. So it's made of a different biomolecule, and its function is not for cell transport as much as it is to give the cell plant its strong structure. This is why plants are relatively less squishy than humans or other animals, right? Like a plant, like a tree trunk, very hard, usually very, very rugged. That's because of the cell walls and other things as well. But like plants needs that cell wall for their structure. Let's see. What's up, Dreary? Happy Halloween to you too, man. Hope everyone had a good Halloween. All right, Ethan, no squeezing mayonnaise into people's bags. That's not a fun situation. What's up, Rachel? What is the connection between diffusion, passive transport, and active transport? The connection between all those is they're all ways in which molecules can move in and out of a cell. The difference between them is, of course, not all of them require energy. Only active transport requires energy. Diffusion and passive transport are energy free. They always go from high to low. They always go away from the higher concentration side. Whereas active transport could potentially go towards the, or will go towards the higher side, which is not the free way. Hey, Tanusha, don't worry too much about ionic and covalent bonds for my test. I'm not going to really go into those on the test. Don't worry about them, Tanusha. Mr. Chang, congrats on the interview, man. What's it for? Is it for a police department or some kind, or what are you interviewing for? What's up, Joseph? Jerry, do bacteria have cell walls? Yes, prokaryotes. Some of them do, some of them don't. Actually, it's a mix. It's a mix. It's a mix. Could I explain the different organizations in a multicellular organism? Tanusha, don't worry about that. I did not go into that. Again, two things that you're talking about are not going to be on the test. Alicia, you said diffusion is like the main term, like a branch, main term, like the branch over passive transport. Yes, passive transport and diffusion are both kind of more related than they are to active transport, although they do also share some similarities. Anna, how are ribosomes and rough ER different? Well, ribosomes are what make the protein. Rough ER is where the ribosomes are located on the surface of the rough ER, and the ribosomes make the protein. It usually goes into the rough ER, and the rough ER modifies the protein once it's been already synthesized by the ribosome. So they're connected, but very different. So basically, when we talk about ribosomes, rough ER, we're talking they're connected in the process of making a protein. Ribosomes come first, they make the protein, and then the rough ER takes it and modifies it, and then eventually will send it to the Golgi apparatus where it's further modified, packaged, and then eventually shipped to its final destination, which might be out of the cell through exocytosis as one option. I think today what I'm going to do is I'm going to try and stay at the front of the chat. So if I don't answer your question, just retype it in and I'll hopefully get it. But I'm going to try to stay at the front of the chat. So if things disappear on me, like behind the chat, and I can't see them anymore, I'm not going to answer them because I can't see them, because I just want to stay more current. I don't like the idea of answering questions that have been asked minutes ago. Sorry about that. Ian, how do we identify which cell is larger than the other? What do you mean? I don't know what you mean by that question. I don't understand it. I don't think I understand that question. Are you talking about like different types of cells? I wouldn't, at least for my test, as far as size and scale of things, I wouldn't ask you to identify which cells are larger than other cells, because I think that's a little too detailed. I think the only thing you should know is that prokaryotic cells, like bacteria, tend to be smaller than eukaryotic cells, that cells that like animals are made of. I got my Erlenmeyer flask today, orange color. Got that Halloween juice. You know what this is? This is, I actually just melted down candy corn. This is just melted candy corn. Which, by the way, can we talk about candy corn for a second? How it's basically the worst kind of candy that's ever been invented. It's basically like little pyramids of earwax. It's the worst candy of all time. And it doesn't even look like corn, right? Like it's not even like. Okay, let's take a peek at this, because it doesn't look like, if you type in candy corn and you give an image, this is not what corn looks like. Corn does not. Whoever invented candy corn, have they ever seen corn? Corn doesn't have like three. It's not a pyramid. It doesn't have three colors. Look, okay, here's candy corn, and then let's look at corn. Corn kernel. Just straight yellow. Completely yellow. So, point proven. What else? Akash, do we need to know the parts of the cytoskeleton? So the cytoskeleton is actually an important part of the cell because it has some important functions. So if you look at, let's see a diagram maybe. This is not a bad one. So the cytoskeleton are these filaments that are made of protein, these long protein filaments that are all spread throughout the interior of a cell. And what it does is it gives the cell structure. It's kind of like scaffolding in the cell. So the cell doesn't always collapse in on itself. It gives it a little bit of structure. And also it turns out that these filaments, some of these filaments are used as like highway systems to move vesicles along. So let's say the rough ER, right, which is right here, has a protein that it needs to transport to the Golgi apparatus, which is right up here. So it's gonna, a little vesicle will sort of form and blob off of the rough ER, and in order to get to the Golgi apparatus, that vesicle has to be transported along the cytoskeleton to fuse eventually with the Golgi apparatus for that protein to be modified further. So in order for anything to actually get around the cell, aside from just diffusing through the liquid cytoplasm, it has to move along these little filaments of protein called the cytoskeleton. So it has two features, two important functions. Josita, we're discussing candy corn because it's ridiculous and I needed to call it out. Let's see. Karan, on the test, will we have to give examples of enzymes and describe them? No, you shouldn't need to do that, at least not for my test. You might need to know function. You definitely need to know the function of enzymes, but I wouldn't ask you, you know, to list as any specific ones per se. Congrats, Mr. Chang. I hope your interview goes well. You'll have to let me know how it goes. So I'm not going to ask you to label parts of the cell. I'm not going to ask you to label parts of the cell in the test. I might ask you to know certain functions of certain organelles, or like I might ask you to know how a protein is produced, right, in a cell. Because again, remember, a cell's main job, to be honest, is just to make proteins. That's its main function, is to make proteins. And so you need to know the organelles involved in the production of protein and sort of the path that a protein might take. You need to know the function of the mitochondria and the chloroplasts and how they're the ones that produce, that convert the energy for the cell to do all these things. Jacob, what are the 4 steps of making proteins? I think you might be referring to the 4 levels of protein structure. The 4 levels of protein structure are, simply put, primary, secondary, tertiary, and quaternary. So this is sort of this is how a protein folds into its final shape, essentially. So a ribosome will put together a bunch of amino acids in a chain. That chain, before it does anything else, we call that its primary structure. Then individual parts of that chain will begin to form pleated sheets and alpha helixes, which are like localized bending of the chain in these 2 different ways. That's secondary structure. Then the entire chain starts to fold into a 3-dimensional shape. That's called the tertiary structure. And then some proteins will then combine with other proteins to have like this multiple protein complex. We call that its quaternary structure, but not all proteins ever reach quaternary structure because not all proteins need to have more than one component. Some of them will just stop at tertiary. So that's kind of like your sequence of events there, from the chain that's formed by the ribosome to ultimately how it folds into a finalized, ready-to-go protein. Yes, Prabhav, one of those A levels that I put in Google Classroom, I'm going to pick one of those. One of those has been chosen for the exam. You'll see one of them on the exam. Hi, Eileen. Nice to see you. Ishan, do you need to know what all the organelles will look like from a picture? No. You do not need to know that. Ethan, what is the function of glucose, glycogen, starch, and cellulose? That's actually a pretty straightforward question. Ethan, I think that was one of the pages of our biomolecule packet. So basically, glucose is a monomer that stores energy for quick use in the mitochondria. Glucose can be turned into starch, glycogen, and cellulose by hooking a bunch of glucoses together. It just turns out that glycogen is a way to store glucose in animals. Starch is a way to store glycogen in plants. And cellulose is used for the cell wall to give the cell wall its rigid structure. MJ, could you explain the process of active transport? The process of active transport, basically active transport uses a protein pump in the cell membrane to pump ions, usually ions, could be other things, pump them against the concentration gradient from where there's already where there's not so many ions to where there's already a lot, like pumping them into a more and more crowded space where they didn't want to flow naturally. So it requires energy, requires ATP to pump those things the opposite direction. Okay, so that's what we mean by, what do you call it, active transport. What else we got here going on? Eileen, you went trick-or-treating? Nice, good for you. Even though you're getting a little bit to that age where it's becoming an issue. This might be your last year, Eileen. Enjoy that candy. Enjoy all that candy you stole. You stole from the 8-year-old little trick-or-treaters. I don't get any trick-or-treaters at my apartment. People don't come up here. Prabhav, will you get an A if you subscribe, like, and turn on post notifications? Can't guarantee it. I don't think there's a correlation, unfortunately. You can't buy your A from me. You need to earn it. That's my philosophy. Let's see, what else? Thanks for the positive words, Kushal. The ones that got through the review. Where else we got? Who else we got some questions going on here? Let's see. Akash, what is the difference between the rough ER versus the Golgi? So the rough ER is the first step after a protein is synthesized by a ribosome, where it helps it maybe fold into its final shape. It might modify it a little bit, and then the Golgi apparatus takes it to the next level and does some other modifying steps. But beyond that, you don't need to know the specific details of the difference between them. Just need to know that the rough ER is first. It modifies the protein, sends it to the Golgi, which further modifies it, finalizes it, and sends it to where it needs to go. But other than that, you know. Ayush, cell walls do not have aquaporins. Aquaporins are in the cell membrane. Mr. Chang, congrats, ma'am. Let me know how. What you have to let me know what college what police department, which college you're doing it for. If I put dry ice in water, is it safe to drink? Yeah, dry ice is basically just solid carbon dioxide. The only problem that you might run into is if you don't take breaths, you might asphyxiate because all the smoke, quote unquote smoke, that vapor that's coming out is basically CO2. So the dry ice it sublimes. It goes from a solid to a gas immediately, and it skips the liquid step. So basically you have a solid carbon dioxide that just turns immediately into a gas. But if you just breathe it in and you don't get enough oxygen, you'll pass out. So you could drink it, totally. Ethan, do you need to know which biomolecule through a diagram? Yeah, I would recommend that you be able to match the biomolecule to a picture of it. You should do that. You should be able to do that. Karan, you're asking, let's say everything is in optimal conditions and there's 10 enzymes and 20 substrates. Is it above or below the saturation point? You wouldn't know, Karan, if it's above or below based on those numbers. I'd have to show you a graph of its activity, and you'd know it's above the saturation point when the activity level peaks off. So enzyme. So if you look at the graph, you'd have to see a graph like this, right? If I just give you numbers, you don't know, because it's like you have no idea how fast the enzymes can work. You don't know. There's no information about that. So you have to see a graph of the rate of reaction. And if it's peaked, like in this line here, you know that that's past the point of saturation. And then if it's still rising, it's before the point of saturation. Later, Ethan. Anna, can I explain the functions of carbohydrates and lipids? So carbohydrates have, their main function is energy, because the mitochondria in all of our cells use glucose, which is a monosaccharide, aka a carbohydrate, a monomer of a carbohydrate, to produce ATP, which is what the cell uses for energy. So pretty much carbohydrates are is where our main source of energy comes from. Lipids, their function is to make up the cell membrane, right? So provide the cell with a structure around its membrane to separate the inside and the outside of the cell. And they also store energy, but for long-term energy, not immediate energy like carbohydrates. So carbohydrates our body will use quickly for energy. Lipids are like something that we can store energy long-term as fat. Okay. Akshay, glycogen is just a polymer, carbohydrate polymer of a bunch of glucoses hooked together. Nice, Amol. Good explanation, as usual. Hey, Shanna. Nice to see you. Renee, what are some factors that might determine if a molecule can cross a cell membrane? So what can determine if something can cross a cell membrane? There's kind of a lot of factors, but essentially I'm assuming you're talking about without the aid of a protein channel or a protein pump, so if it can just diffuse through the lipid bilayer, right? So if it can just diffuse through the lipid bilayer, it has to be small enough to squeeze in between those lipids, and it has to be hydrophobic, meaning repelled by water and attracted to lipids, right? So it has to, because to get through a lipid bilayer, it has to be able to be small enough to squeeze through, but it also has to be able to pass through this layer of lipids and not be repelled by it. So it has to be hydrophobic. So small and hydrophobic. Ishan, is the function of nucleic acids just for DNA and RNA? Yeah, nucleic acids, their main job in the cell is just to store genetic information. And when we talk about genetic information, we mean the instructions for making proteins, right? So proteins are what do most of the functions of the cell. They're I can't say most important biomolecule because a cell can't survive without all four, but they have the most different variety of functions. They do a lot of different things. And the instructions for making those proteins, like the blueprints for how to make them, that the ribosome uses to assemble the protein, that information is stored in the form of nucleic acids in the nucleus in the form of DNA in most eukaryotic cells. MJ, microtubules and microfilaments are the two types of proteins that make up the cytoskeleton. No problem, Renee. Priyanka, don't need to apologize for being late. There's no, this is just an open stream. Open stream. Come anytime. Quran, unsaturated means a fatty acid chain with double bonds. Correct. Correctamundo. Akash, what factors determine whether something is hydrophilic or phobic? If it's what determines that is if it's polar or charged. So if a molecule is polar or it has a charge, like a positive or negative charge to it, it's going to be hydrophilic. It'll like water. Molecules that are not charged or non-polar, those are hydrophobic and they prefer to be near lipids and away from water. You don't need to know the names of the essential amino acids. Just know that they're the ones that you have to eat in your food because your body doesn't make them. Therefore, they're essential in your diet. Varshini, or Varshini, what does selectively permeable mean? It means it only allows certain molecules to pass through, which Amal just basically took the words right out my mouth. Yeah, it only allows certain molecules to pass through it. Ishan, that's a big question as far as the differences between prokaryotic and eukaryotic cells. And I'll just put up like a Venn diagram and you can look at it while I answer other questions. How about this nice one with Dora the Explorer on it for some reason? I don't know why. Well, no, the resolution is very small on that. It doesn't look too good. This is probably a decent one. Also, the resolution not great. You guys can't see that maybe. Here we go. There you go. Take a look. That's kind of the big ideas there. Renee, bulk flow is the same as facilitated diffusion, right? Bulk flow is a term used for where most of the water, how most of the water moves across the cell membrane, and the bulk of water does use facilitated diffusion through aquaporins. So they're not exactly the same thing, but bulk flow uses facilitated diffusion. Hey Maggie, thanks for joining in. What are motor proteins? Motor proteins are basically these proteins that move along the cytoskeleton to transport vesicles carrying a variety of different things. So we talked about how vesicles are these little bubbles of membrane. They move around the cell. The only way they move is by being carried by motor proteins. I can show you a picture of a motor protein. So here's a very simplistic diagram of a motor protein. It's this guy right here. So this is like the microtubule, this is the cytoskeleton, and this motor protein sort of walks along the cytoskeleton carrying a vesicle above it that has like something inside of it. There's some good videos on motor proteins that you could watch online if you're interested. So here's like actual image, not an actual image, but like a three-dimensional image. So this is inside the cell. This is your motor protein right here, and this big blue bubble is a vesicle, and this motor protein is just absolutely jacked, and he's just able to carry this giant vesicle just all the way along this motor protein, or sorry, along this micro filament right here. No problem, Renee. Anytime. Karan, yeah, long-term energy storage are one of the functions of lipids. Their second function is to make up the cell membrane, compose the cell membrane. Prabhav, you're eating candy for dinner? I guess I'll excuse it. I'll excuse candy for dinner on Halloween. It's a good one time a year where it's acceptable to eat candy for dinner. You're getting your glucose. You're getting your glucose, my man. Going heavy on the carbs. What fills the inside of a cell and allows movements of materials within the cell? That would be the cytoplasm. Ian, it could be the cytoplasm for diffusion of molecules throughout the cell. It's like a liquidy filling. Or it might be the cytoskeleton, which allows for the movement of things along that as well. MJ, lysosomes. Lysosomes are basically an organelle in the cell whose job is to basically recycle or break down unneeded waste material that the cell, right, like old proteins or molecules that are no longer needed anymore. The lysosome basically breaks them down and recycles the material. Ian, why do enzymes work better at certain temperatures? Because the shape of an enzyme, its shape, will only hold together at the right temperature. So if it gets too hot, the shape starts to change. And if the shape changes, the function changes and it can no longer work as well. So enzymes only work at certain temperatures because their shape is only able to hold together at those certain temperatures. Anna, yes. Anna, cytoskeleton and cytoplasm are different. The cytoplasm is the liquidy filling of a cell, like the liquid that everything is in. The cytoskeleton are these filaments, these protein strands that give the cell structure and allow things to move along them to be transported. What's up, Akash? How you doing, man? You got 25 pounds of candy I used from half hour of trick-or-treating? How? How did you get that level of candy? That's insane. Do you live on like Willy Wonka's street or something? Pre-writ, lipids only have subunits, not monomers, right? That is correct. Lipids only have 2 subunits. They do not count as having monomers. Ian, what happens to an enzyme when it's too cold? The enzyme will probably slow down its function. It probably is less likely to change its shape. Usually only heat will change the shape, but it's possible it could lose its shape a little bit, but it just will work slower. It'll just work slower. And when it gets too cold, it'll just stop working. It'll just sort of freeze. Akash, do prokaryotes have ribosomes? Yep, they do. MJ, I've answered that question before. What does it mean for a cell to be selectively permeable? Basically, it means that a cell is selective, right? So it's not everything is allowed to the word to get into it or out of it. So permeable, the word permeable means things can move in and out, right? Right. So a good example like that you might be able to connect it to is a cell membrane is selectively permeable, but so is something like a screen door or a window screen. It's selectively permeable because air can get in and out through a screen, right? But a bug can't because it's too big. So the cell membrane is selectively permeable in that sense that some molecules can get in and out, but others cannot because they're too big or they lack the right characteristics to cross. You take 5 candies from a bowl that says take 1. My man, you're stealing. You're a thief. What else we got here? Drary styles. Are there proteins that can function at very low or high temperatures? Yes, there are. There are certain proteins that are adapted for that. Like if you think about, there are certain fish that live in very, very cold waters. They have proteins adapted for that. There's also organisms that live at very, very hot temperatures. Their proteins are adapted for that. Prabhav, how does pH affect enzymes? In a very similar way. If the pH gets too high or too low, the enzyme's shape changes. Just, it's very similar. Very similar. Karan, what are the functions? Prereq, when an enzyme shape changes, so does its function, right? Yep. Enzymes shape and function are connected. What are the functions of proteins? Karan, there's many different functions of proteins. There's a lot of them. Just you can look that up online. Just simply type in functions of proteins and you'll see that it's a variety of different things, right? They catalyze reactions. They are involved in cell transport, moving things in and out of the cell. They're help to fight off infections and with our immune system. They make up your muscles, so they help organisms move. Renee, smooth ER synthesized lipids while rough ER synthesized proteins. That is correct. Ankita, what is a lipid bilayer? A lipid bilayer is the cell membrane that's composed of 2 layers of lipids. So it has 2 layers of lipids. So if you look at the lipid bilayer, this is a lipid bilayer because it has 2, it's a sandwich. It's got 2 layers, right? The top and the bottom. Double layer. Ankita, glycoproteins, those are proteins that you would find on the cell membrane that have a protein and a carbohydrate stuck together. So it's got like 2 biomolecules combined into 1 molecule. So that's what that is. Brianna, the phospholipid bilayer and the lipid bilayer are the exact same thing. Mary Jane, how do enzymes help chemical reactions occur? They basically speed up the reaction by providing a little bit of a boost in terms of the positioning of the molecules so that things align correctly, and they just make it easier for it to take place. They facilitate it. How are proteins formed? And can I show you a diagram? How are proteins formed? You don't need to know that yet, Quran. That's something that we're going to get to at the beginning of the second semester, because we're going to talk about protein synthesis at the second semester, and we'll have a whole topic on that. We'll talk about that for a couple weeks. Pre-writ, what is the function of the Golgi apparatus? It's sort of the packaging center of the cell. It's what, if we have like a finalized protein that needs to be shipped somewhere, the Golgi apparatus packages it up into a little vesicle and then sends it off to the cell membrane to be released to where it needs to go. Ishan, you are correct. You are correct. Isotonic is when it is the same and water goes both ways. So you have the same concentration on both sides of the membrane, so water moves in and out equally. There's no change in overall movement of water. Gosh, Minecraft later? I don't know, man. I don't think tonight is not gonna happen. I'm just way too tired tonight, to be honest. Tonight is not a night for me to stay up too late. Maggie, you're from the apes class? Don't mind you? Okay. You blend in. Just blend in. Just blend in. It doesn't matter. You can be here. You're all good. Everyone's welcome. If you have a question about apes, fire it my way. Ian, no. Long-term energy is not related to proteins. Proteins are not used for energy. Angela, do you need to memorize all the organelles and their function? I would say it's a good idea to know the organelles and their function. Yeah, you should know what they each do. I won't ask you about all of them, but I'm going to ask you about some of them, so you got to know them. Well, Mr. Chang, I am also injured at the same time. I have I had my ACL surgery a couple weeks ago, so I am injured. So I have two excuses. Pre-writ, how do cells communicate with each other? They can send messages through things called hormones. So one cell can release a chemical hormone that travels to another cell to send a message. What about second birth? It's a good question, Brianna. What is my favorite organelle and why? That's a tough question. My favorite organelle. Never considered it. Never considered that question. My favorite organelle and why? Yeah. It has to be the chloroplast. It really has to be the chloroplast. Because it's what starts the whole thing. You can't get going without the chloroplast. It starts the whole food web. Because it converts light energy into chemical energy. Like, that is this amazing feat. right? Without chloroplasts, there would be nothing else, because it's the only thing on Earth, biological thing on Earth, that can take a photon of light or photons of light and convert them into sugar, right? Glucose, that the rest of the food chain uses. The only energy available to us on Earth, mostly, is coming from the sun. If you didn't have a chloroplast, that would never be able to be transformed into other forms of usable energy. So you got to give big ups to the chloroplast. You really do. I mean, all that candy you guys are eating right now, without that chloroplast, you got no candy. That's sugar. Plants made that initially. Yeah, the chloroplast gets my vote, big time. Jerry, what about the mitochondria? Mitochondria it's the powerhouse of the cell, but people don't understand. Without the chloroplast, the mitochondria's got nothing, because the mitochondria takes glucose and turns it into ATP. Where does that glucose come from? Our boy, CP, chloroplast, our green guy, our green boy on the other side. Without him, the mitochondria's got nothing. So mitochondria gets is overhyped. The mitochondria is overhyped, so it goes to the powerhouse of the cell. Yeah, but where's all that power coming from? It's coming from the chloroplast, who's doing all this work under the radar. Let's see. What's up, Blue? Happy Halloween, man. My Halloween costume, I had 2 Halloween costumes this year. My one that I did at school with the rest of the teachers was a mad scientist. So I had a lab coat with that had like explosion on and that kind of thing. But I went with my friends to do a Halloween thing, and my costume was I was the final lap guy from Mario Kart. So that costume took me a long time to make. I spent probably like 10 hours making this final lap guy costume. What's his name? Lakitu? This guy? This guy. This was my costume. I went as this dude. Let me see if I have a picture of my costume for you guys. Here, this was the costume. That was my Lakitu costume, final lap. That cloud took me like forever to make. And then the final lap sign was decent. It took me a while. You can't see my back, but I made a turtle shell for the back as well. So that was my Lakitu costume. What other questions? It's 8:55, so we're kind of coming down to the end here. I don't know how much longer I'm going to go. Questions are kind of trailing off. Maybe people are getting their sugar crash. They've already kind of maxed out all of their, what do you call it? Glucose levels in their blood has plummeted. Okay, there we go. Some more questions. Next year I should be a Goomba, Akash. How would you make a Goomba costume? It seems like it'd be very clunky. I'd have to have a big, like, brown thing over me. Let's see. Karan, what are two types of nucleic acids? RNA and DNA. DNA is in the nucleus. RNA you typically find in the cytoplasm, floating around. What else? Renee, you're lactose intolerant, so that means you have a lack of enzymes. Yeah, you're missing the lactase enzyme. Mary Jane, what does it mean that all cells come from pre-existing cells? That's because the way more cells are made is by mitosis, which is the cell division. Right? So one cell divides into another cell. So in order to have a cell, you have to start with a cell. Ian, do proteins just make stuff in the body and use as enzymes? Proteins don't make stuff. Well, they kind of make stuff, but proteins have a lot more functions than just those two things. So they have a lot more functions. If you just type in protein functions, you'll see a list, and it's pretty reliable. Nah, can you explain polyunsaturated fats? Polyunsaturated fats, that's a kind of lipid or kind of fat that has multiple double bonds in it. So that's pretty much it. Unsaturated fats tend to be better for you than saturated fats on average. Akash, yeah, there's lactase pills. You can take a lactase pill when you eat dairy to break down the lactose. So some people do that. Ishan, like I said, unsaturated fats are typically more healthy for you than saturated fats. But that's arguable. It's been argued that maybe not, but it depends. It's a complicated topic. We're going to get into that more at the end of the year when we talk about nutrition. Karan, what is the difference between passive transport and diffusion? The difference is that diffusion does not require a protein. Passive transport requires a... Oh, actually, no, never mind. Diffusion is a type of passive transport. We'll just leave it at that way. Hey, Nerea, what exactly is an active site? The active site is the location on an enzyme where the actual reaction takes place. So let's look at an image. Maybe of an enzyme might make it better. So if you see on this image, the active site is this location here, these little grooves on the gray blob. So the gray blob is your enzyme. This is the active site. What the heck? That's the active site. That happened right there. And the substrate is this green molecule. So what happens is the substrate will bind to the active site. It fits perfectly in there. The enzyme then catalyzes the reaction or causes the reaction to go forward, which looks in this case like it's splitting the two parts of the substrate, and you get your products. So the substrate goes in to the active site, binds, splits apart, and is released, and then the next one can come in. That's what we mean by active site. Nah. Can you explain facilitated diffusions? So facilitated diffusion is basically just when a molecule is too big or too hydrophilic to go through the membrane through diffusion. So it needs a protein channel or a protein transporter to bring it across or in and out of the cell. So it's basically just diffusion with the help of some sort of protein to get it in or out of the cell. Karan, the difference between protein channels and transporters. There's a difference, but you don't need to know it. Not for my class, and I'm not going to explain the difference because it's not necessary. They're both just used to bring molecules in and out of a cell that can't just do can't go through the membrane directly. Good one, Brianna. Channels are where YouTubers reside. David, this is the worst week of your life? That's a bold statement, man. You've been alive for a lot of weeks. This is the worst one? What about the week of your life where you were born? That must have been a pretty traumatic week, man. To go from a warm, cozy environment of the womb, just floating in embryonic fluid, to just being ejected into the harsh, cold world. That's a rough week, man. That's one of my least favorite weeks of my life. How is it pronounced, Prabhav? No, like N-O-O-H, like no. So it's not na, it's no. Well, it's definitely not nah. I didn't say nah. I said na, like N-U-U-H. Like how it's spelled, like N-U-H. So is it no? I don't know. I don't have that person in my class, so. You don't remember, David? Okay. That's fair. I really don't either, but I just imagine it was a rough week. Whenever I'm having a bad week, I just think about what it would have been like to be in that week. And then I go, okay, it's not too bad. This week, it's not too bad. This week's okay. Ayush, you got 2 tests and an archery class tomorrow, and an archery tournament at Berkeley the day after tomorrow. Ayush, you do archery? We're trying to get an archery club up and running. If you're interested in joining, let me know. Oh, it's like no. How long do you, but how many O's? Is it no, or is it no? How long do I hold it? That's my question. Like the new, you in new? So the you in new? There is no you in new, Amol. You as in poo? So it's new. So it's basically pronounced new. New. Okay. You just say new. If it's pronounced new, you just say new. Don't say poo with an N. I mean, all right. New. I got it. It's new. I'm on top of it. See, that's all you got to say. I know how to say new. Okay. Okay. So where are we at now, everybody? How are we feeling? We're at 9:06. Covered a lot of different topics here. What's up, Amy? I'm coming in right at the end here. How do cells regulate the movement of molecules in order to maintain homeostasis? So the way they do that, Amy, is they use their cell membrane to regulate what comes in and out of the cell. So they can select which molecules come in and which go out. Right? So in order for a cell to maintain its internal conditions, like that's what homeostasis is, right? To maintain the internal inside of a cell the way it wants it to, it can't just let everything rush in or everything rush out. So its membrane protects that from happening, right? So it can allow certain things in through protein channels, like facilitated diffusion, or protein pumps for active transport. Right? So it's regulating what comes in and out in that regard. So that's how it maintains homeostasis. Does that make sense, Amy? Renee, I like how you said, I hope you guys do fine on the test. Like you didn't say like, I hope you guys do like good or I hope you guys do amazing. You're like, I hope you guys do decent on the test. I hope everyone does okay. They do fine. We'll do fine. Karan, is it possible for two substrates to react together and become one product? Yes. So this picture I have here shows one substrate becoming two products, but you can have a reverse reaction exactly the same. You could have two products come in, get connected together, and then go out as a single product. Or sorry, two substrates come in, react together, and then it'd be released as a single product. That's possible as well. Possible as well. Jeevith. Jeevith Christ. Why does sugary and salty environments kill bacteria? Because think about what happens to a cell in a very hypertonic solution. All of the water rushes out of it and it shrivels up. So if you expose a cell, like a bacteria cell, to a really, really sugary or really, really salty solution, all the water rushes out of it because water will move from the inside to the outside where there's more stuff and the cell will shrink and die. That's why, just this is a little fun fact for you. This is why when we have salted meats, right, or pickled things, what we're doing is bacteria does not grow on a very salty meat. So the way we used to, before we had refrigerators, to stop bacteria from growing on our food and causing it to go bad and go spoiled, before we had refrigerators, what they would do is they would just coat it in sugar or salt, because bacteria cannot grow in such sugary and salty conditions. So, you know, salted hams, all our meats, we would make very sugary or very sweet on the outside, coated on the outside to protect it. It's a little bit of a little pro tip for you, a little history there. Let's see, any other questions before I leave here? I think I'm gonna call it a night pretty quick here. It's getting close to 9:15. This is a short stream, I think, just because I'm particularly tired for some reason. So yeah, I might call it a night, but let me take a peek at some of the questions I missed. I don't know if I missed any. You guys are just talking about the likes, how many likes it has. You guys, it's okay if you don't all like my video, because I don't judge. I don't base my self-worth and my happiness on the amount of likes I get. You know what I mean? It's okay. You can let that go, Mr. Chang. You don't need to review that. Mr. Jones said guaranteed A if you like, subscribe. You got to 15. There you go. Appreciate it. Appreciate you guys. I think we're testing it up here. I thought I saw another question from Nerea, but I lost it in the chat. Nerea, what was your question? Here it is. For the structure of plasma membrane, what is a phosphate head and a nonpolar tail? Nerea, take a peek at this diagram. This is the plasma membrane. The phosphate head are the pink circles. The nonpolar tail is the blue part. So the nonpolar tail is one part of the lipid, and the phosphate head is the other part. Those are the two subunits of a lipid. Okay. What else? Did I see another question? Some of the questions you guys ask, like a mole, you ask, like Ayush and a mole, you guys sometimes ask really cool questions that I wish I could spend more time talking about. Like, can we simulate touch through false neurotransmitter? The answer is probably yes, actually. You guys are like thinking at such a high level. Correct, Ayush. The head and tail are the subunits of the lipid. That is correct. Quran. I can't talk about all the similarities and differences of a prokaryotic and eukaryotic cell. Although, did I have that up at one point? Where did I have that one? No. There you go. Boom. Got it. There you go. Ashley, what specific enzymes do we need to know? You don't need to know any specific enzymes, Ashley. I wouldn't quiz you on that. Just trying to make sure I didn't miss anything. Okay. All right. I guess I'm just going to answer this one last question from Ian. Why is it bad for your cells to be shriveled or swelled? Because if they swell, they could eventually burst, and they might, that would mean they would die. And if they shrivel, if they get too small, everything inside is going to be squished into one small area, and it's probably not going to be so good for the functioning. It needs the right amount of space and structure. Yeah. All right, guys. I'm going to let this go a little early tonight. I appreciate you all. It wasn't a too crazy night. Like, I think we peaked out around 40, 45 people, which is good, because I remember sometimes before we were getting up into the 70s or 80s, and it was getting chaotic. But yeah, I think today wasn't too bad. Kind of half of that number, which was pretty good. So yeah. All right, guys. I think you're gonna do fine. Don't stay up too late as usual. Be reasonable with your sleep. Hopefully you guys aren't all jacked up on Halloween drugs like candy corn and M&Ms and Laffy Taffy, and I'm trying to think of all the candies that you used to get. It's the same. Candies are pretty much the same as they've always been. They don't really change too much. But don't overdo it tonight on that. Save some of that for tomorrow. Save it for the weekend. Okay? Look forward to that, to your little stash. Back when I would trick or treat as a kid, you'd get a pillowcase full. It would last you months. I think I still have some candy from when I was about 10 years old, just lying around. Might go treat myself to a 20-year-old mini Snickers. I think that'll probably be tasty. So get some sleep, eat a good breakfast, enjoy your night. And yeah, Priyanka, that's exactly the advice I would give. Eat breakfast and get good sleep. Bingo. Bingo. So I'm going to leave you with that. So enjoy the rest of your night, guys. I will see you all tomorrow. Actually, I won't see you tomorrow because I'm not going to be in class. I'm going to try and sneak out during tutorial, though, so I may be in my room during tutorial. That's my hope, unless the rest of the bio team doesn't let me sneak out. And if I made an appointment with you, I apologize because I forgot that I was going to be absent or gone that day. But anyway, peace. I'm out. Bye, everybody. Have a good night.